everybody.
Good evening. Welcome to AI Live here at the Aesthetic Immersion. We're live in Brea, guys.
We are. We have a new set. If you notice, it's a little bit of a different set. So our team is being more creative. So it kind of feels open. I feel open and airy
I
here. It's
know. I feel
awesome.
that the lights are making me look shiny.
I know, me too.
No, this is all the PDRN
It's
and
all your
PN I've been doing.
topically.
Of course, the microneedling. No, this is
Well,
all the Cabo sun I got.
I love it. He was in Cabo last weekend. So not fair. He got a little color in the sun. I love it.
Trying.
Well, welcome everybody. We're so glad you're here with us.
So today we are going to jump into an exciting topic. So in the world of regenerative, biostimulatory, bioregenerative, this is one of the cool products out on the marketplace. They are based in out of science. We're gonna bring in plated.
Plated, yep. So ththese are platelet-derived exosomes. Dan Harrison is an integral part of that team. And so we kind of wanted to have Dan, we want you to kind of educate us on plated, how platelets derived, how these platelet cells are, how these exosomes are different, how
Yeah.
these are different, how they're harvested, all this good information that you can provide for us about this product.
Thanks, Dan.
Thank you.
Yeah. Thank you, Gideon. Thank you, Lori. I appreciate you having me today. I've been looking forward to this for some time. And I think the intro was sort of perfect. You could see the passion and you seemed to get excited about sort of innovation. And
Yeah.
let's face it, in the US aesthetic space, innovation is the lifeblood of makes all this tick.
Right.
So whether you are a manufacturer, like, I work for a manufacturer, or if you're a patient or an aesthetic provider, we're always sort of looking or on a quest for better solutions to this, dastardly aging problem or signs of aging, aren't we? And,
Always.
yeah, that's what's appealing about the aesthetic space. But if we're honest, if we look at sort of the topical category and the skincare category, it may have lagged just a little bit in terms of innovation compared to some other sectors. And that's why we're so excited about what we have here in Plated Skin Science. It's a true human-derived regenerative product that contains exosomes. So really excited to kind of jump in a little bit into the story and answer any questions that you or your network has this evening. So thanks you so much for having me.
Thank
Awesome.
you.
Thank you.
Okay, cool. I think we'll pull up a deck here in just a second. Awesome. So this is the product, and this is what we're so excited about. Plated Skin Science is the result of 2 decades worth of research and development at a pretty esteemed academic hospital, system up in Rochester, Minnesota. And it's a true regenerative product. And you touched on this in the intro. This category, regenerative medicine or regenerative aesthetics, it's certainly a big buzzword. It has been for some time. And I think a simple definition or description is sort of leveraging the body's ability to renew itself, ideally on a cellular level. And there's a lot of fantastic products out there, a lot of products that do a nice job, but the bulk of products out there on the market they're either chemical agents or botanicals, right? And when you talk about renewing on a cellular level, there's some limitations there, if we're honest. So that's what's so cool about this. This is a true human-derived product. There's no substitute for human-derived, we think, when you talk about renewing at that level. There's no lab engineering here. What our company has been able to do is simply separate an exosome from a human platelet in its native state. So with all the goodies that contains and the sort of naturally been sort of put into that exosome, and we have it in a room temperature shelf-stable serum called Plated Skin Science. So, it's been out for about 3 years, and we're in a few thousand clinics in the US, and tens of thousands of patients are using this on a regular basis. So we'll get into the science and the data here in a little bit, but the sort of the repurchase rate, the reorder rate from our clinics is something that I haven't seen before. And I've been, in aesthetics and dermatology for, my entire career. And I think it's just validation of sort of the efficacy and how this product works and those true sort of natural results that we're looking for, as, you kind of mentioned in your intro. So I'll jump in if you don't mind, and again, answer any questions that you have, if that's okay.
Beautiful.
Okay. So let's do just a little bit of sort of 101. When we get on, to these sort of webinars, there's a varying degree of understanding of what exosomes are. So we'll just sort of ground our session today, if that's okay. So what are these? Exosomes are bioactive nanoparticles that are naturally produced by our bodies. So bioactive in that they're not alive, they're acellular. They're simply transmitting a message. This is what cells use to transmit a message to another cell to get that cell to act in a certain way. So think about, like, if you want a visual, like a message in a bottle, that's all these things are carriers for a very specific message. So the right exosomes can promote regeneration and renewal. And that's what we're looking for here. Again, if you think about the definition of regenerative medicine, leveraging the body's ability to heal or renew itself, this is a very special sort of breakthrough, in this regenerative aesthetics category. I think this slide is really important because, these things have rushed onto the scene in recent years. And it's important to note that not all exosomes are the same. So they carry varying messages, and not all of them are great messages. So all cells are emitting these exosomes to communicate with other cells. Sometimes they communicate things like inflammation or metastasis or cell death. Sometimes they signal tissue renewal. And a lot of this messaging hinges on where these exosomes come from in the body. It's very important. So our team, over the course of 20 plus years and tons of research, again, up at Rochester, Minnesota, using all sorts of different sources, we sifted through and we landed on a very unique source for our exosomes, which produced a high amount of these tissue renewal exosomes. So again, just because you see the word exosome on a bottle, it doesn't mean it's transmitting that tissue renewal message. And that's very important, I think, for the listeners today to understand. Are there any questions on this, Lori or Gideon?
No, very simple.
Okay. So we utilize human platelets. This is our source. And in the US, there's a couple of camps of exosomes. There's mesenchymal stem cell or stem cell derived exosomes. And that's the bulk of the companies in the US. We use human-derived platelets. And we found that this is the most ideal source for consistency of that tissue renewal message. And it makes a lot of sense. Platelets, as we know, are the first responders to wounds. And these platelets shed that very specific exosome that signals renewal. So we get a lot of questions usually at this point, like whose platelets are they? Where do these come from? So we have contracts, our company, with transfusion grade blood banks across the US. And we collect these platelets every couple of weeks from multiple donors. We take them up to our facilities in Rochester and begin our biomanufacturing process. So who are the donors, right? Who are the donors? That's a very, very common question. Again, this is pooled. So we use multiple donors, for each sort of batch of plated skin science. The nice thing about using the US blood banking system is it's so tightly regulated, right? These are platelets that are used in massive transfusions on a regular basis in a hospital setting. The benefit of using a pooled approach is you're not banking on one patient to, sort of fuel a product line or a company. We collect these again every couple of weeks. They're pooled together. And the nice thing is we're playing the law of averages with our source. So some of the donors may be producing, fantastic amounts of real healthy platelets that are producing great exosomes. And maybe you have some patients that aren't doing it, producing as much. But when you pull them together, you sort of take out the vagaries, right, in an odd way. And what you're ensuring is that you're getting a product that's firing and acting, within very specific parameters from a renewing standpoint. So the platelets are vetted for all sorts of bloodborne viruses that you would expect, perfectly safe and pure. We transport them up to Rochester, Minnesota, and then they undergo further testing before this manufacturing process. So are there any questions on this?
Is there an age that you look for, or is there an age criteria?
Yeah, so Laura, it's a good question, and we don't use geriatric patients here. So it's a bit of a younger demographic, certainly not like 75 or 80-year-old patients. But interestingly enough, sometimes it's not the younger patient that is producing, the best platelets or exosomes that are designed to regenerate. Younger patients may not need regeneration as much as an older patient. So it's an interesting twist on that question or that assumption. Thank you for asking that.
Interesting.
Yeah. So let's take a closer look at the anatomy of a regenerative exosome. Again, they're not all the same. So I'll point out just a couple of sort of, characteristics of a good regenerative exosome or a platelet-derived exosome. So you have these very unique surface proteins that promote the uptake of this exosome into a cell. And that's the beauty. We talk about healing from within. Well, that's at a cellular level. So a good exosome is taken up into the cell, and what it does is it sort of hijacks the endosomal compartment of that cell and programs that cell to act in a certain way. It has a lipid bilayer that's very important. We'll get to that in a little bit more detail in a little bit. But that lipid bilayer protects the cargo and provides stability of that exosome. And if that's cracked, or if it's ruptured in any way, there's potential to render this biomolecular cargo unstable. And in effect, your exosome is not, producing the results like you would want it to. And then lastly, maybe most importantly, what is that biomolecular cargo? So a platelet exosome is loaded with phosphoproteins or phosphopeptides, antioxidant enzymes, and some nucleic acid as well. So these phosphopeptides or phosphoproteins, what they're doing when they hijack the brains of the cell, they set off this cascade of renewing signals throughout the intracellular pathways in that cell. Again, getting it to act in a certain way to renew. The antioxidant enzymes are really unique. If you compare this action, for instance, to like a vitamin C, as we know, a vitamin C is picking off free radicals once they've left the cell, typically in like a one-on-one fashion. The antioxidant enzymes in an exosome, once this exosome gets into that cell, are shoring that cell up against the release of radicals, or it prevents the production of radicals. So we're catching it much earlier in the sequence, which is really nice. For an aesthetic patient, the benefit of an exosome is it drives pro-collagen formation, pro-elastin formation, and then overall, it's normalizing the activity of that cell, ensuring sort of proper turnover of that cell, which is really nice. So that's what this is doing, again, from the inside out, which is really cool, and why this is such an advance in this regenerative aesthetic space.
I have
I'll
a
pause
really
there.
stupid question.
Yeah, of course.
Might be a really stupid question.
Not at all.
Nucleic acids, are we looking at the biomolecular cargo, the nucleic acids, are we looking at maybe mRNA that those are?
Yeah, microRNA,
Okay.
RNA, excuse me.
Okay.
And I think, Lori, we'd had a conversation once about, the COVID vaccine, for instance, right? And, people may be curious about
That's where my
that.
brain's going. Yeah.
Yeah, exactly. We've had that before, and it's a really, really good question. So it's highly unlikely that that gets taken up by an exosome for a couple of reasons. We know that the lipo nanoparticles in that, they target local immune cells like myoblasts in the liver. And it's those local cells that, express the spike protein, and then taking it to the spleen for memory B cell generation. So they're not taken up by exosomes. And the second reason that, we're not concerned with that is because, a platelet has a pretty short half-life, right? It's less than a week. So, if you, for instance, got a vaccine and then, you donated blood, it would have to be like a remarkable sort of, timing there. Because these in the blood banking system are used like pretty quickly, it's not like a 2 or 3 week thing, it's a week. So hopefully that answers your question. It's a very fair question for sure in this day and age.
Sure. Okay. Thank you.
Yeah, you bet. I will just on these surface proteins on the left, I should have touched on this. So there's a couple of very unique surface markers here, CD9 and CD63, which categorize this not just as an exosome, but a regenerative exosome. Again, they're not all the same, these things. Yeah. I think the size is also very important. Oh, goodness, let me go back one, sorry. They're very small, right? So under a couple hundred nanometers. Actually, most are like less than 150. But we always get asked because this is designed as a topical product, for skin aging and a daily take-home treatment. So how does this get where it needs to go? So very, very tiny, about 150 nanometers. If you think about, a hair follicle, or a sweat gland opening, I think those are like 30 or 50,000 nanometers, sometimes bigger. So, they certainly get into the skin. But also they act like homing pigeons in a way. Think about platelets and how they work in your body, right? So they go to where they need to go. And that's the same sort of concept with a platelet-derived exosome. This is a very refreshing slide to our clinics. So I'll take you through just quickly the timeline of the development of this technology. Again, exosomes have rushed onto the scene, right, in the past couple of years. There's so many companies out there now that are talking about exosomes and selling exosomes. And, if you're a clinic or a provider and you're entertaining, bringing them on or, which one you're going to bring on, it's kind of hard to decipher, which way to go. A lot of our clinics find this very reassuring. And I mentioned this earlier, that this has been in development for 20 years. And our founders are a team of cardiologists and PhD scientists that run the regenerative medicine program at a very prestigious academic hospital system up in Rochester. And they spent their life's work studying stem cells. In fact, one of the largest stem cell trials ever done on cardiac patients in the US came out of this program up in Rochester. And one of the key findings from that program was that, when these stem cells were working on cardiac patients, it wasn't actually the stem cells. It was this very unique biology called an exosome that was leading to soft tissue regeneration. I mentioned these guys were PhD scientists as well. So they studied the lots of stem cells that were working. So that was the big first breakthrough for the company. The second was landing on platelets as an ideal source. Again, there's varying sources in the body. We spent years and years trying to make stem cells work, but there's some inherent limitations when you use a stem cell as a source. They're alive. You want them to go left, they go right. You want them to go up, they go down. And there's some sort of lab components when you utilize a stem cell that can lead to some inconsistency. So at least for our team, when we moved away from stem cells and landed on platelets, all those inconsistencies in terms of performance got put to bed. So that was the second big breakthrough, landing on platelets. And then lastly, and perhaps one of the trickiest components of what we do, is on the biomanufacturing front. So how do you get your exosomes from your source material, take them out in a stable fashion, maintaining viability, and an ability, the ability to produce these at scale. Traditionally regenerative medicine can be very, very expensive, but this breakthrough on the biomanufacturing front was really nice, and it unlocked several doors for our company. And one of those doors was the development of renewal technology, which is the cosmetic version of our platelet-derived technology. We have a massive medical side to our company that is in, FDA approval process for varying applications, including wound care, degenerative joint disease, a cardiac application. And so we're sort of this little bitty, startup cosmetic division, which has come to the market with plated skin science about 3 years ago. So I mentioned, the uptake here in the past 3 years has been tremendous. The product is, it's been so successful, and there's been a lot of accolades and a lot of awards. Perhaps one of the most exciting ones for the team was Best Inventions of 2024 from Time Magazine of all publications, which was really cool and nice validation of our technology. So the team was really excited about this. It came at the end of last year. We were the only skincare product to win this award, which was really cool. So. I'll come back and touch on this biomanufacturing piece because when it comes to exosome manufacturing, this is it's one of the trickiest components of the whole thing. A lot of companies use a sort of high G-force, aggressive spinning, process to separate exosomes from their source material. And probably a good analogy or a real world example is PRP, for instance, in an office, which sometimes can render, excellent results for sure. But one of the things that we know is these exosomes can be a bit fragile. And if that lipid bilayer is compromised at all, it can lead to instability, of that cargo. So we separate our exosomes from platelets in a very gentle manner, again, maintaining quality and purity. And the nice thing about this process, and what we think is a real competitive advantage, is that it's room temperature shelf stable, so no refrigeration necessary. Another analogy we use is like the egg analogy, like a chicken egg. So for instance, if you have an egg that is cracked, you probably better use it fairly quickly. If not, it's got to be in the fridge, But if that chicken egg is intact and there's no cracks at all, you can leave it on your counter and preserve it use by date, maybe 2 or 3 days later. It's fine. So that analogy holds true with exosome manufacturing. So this is a really cool, advantage to this technology, which offers a lot of, practicality for sure. I'll pause there. Are there any questions, Lori and Gideon, on this? Okay. So just as a summation, we use platelets, which in the body is actually the most prolific producer of exosomes out there. When you combine that with our biomanufacturing process, we're able to produce massive amounts of these exosomes. In fact, each bottle of plated skin science has approximately a trillion of these regenerative exosomes, which is nice. So every pump, up to 15 billion per pump. And this is designed, as I mentioned earlier, as a take-home treatment for daily use. So that's where this is very important. There's a bit of a misnomer about exosomes in that you can use them in like a one-shot fashion in a chair in an exam room. So all of the research, that we've compiled over a couple of decades suggests that you can't do that. Your cells need to interact with this messaging on a regular basis, in order to go into that pro-collagen, pro-elastin formation mode. Analogy here is, say you had a young child and you told them to, go up to their room and make their bed. If you told them once, it probably is not happening. But if you told them like 10 or 12 times, it starts to happen, and then you get that behavior. Kind of an odd sort of example, but it holds true with that sort of need, for repetitive use of an exosome.
So people are using this after laser resurfacings, after microneedlings, things like that. What's your
Yeah.
opinion on that?
Yeah, it's fine to do that, Lori. I think when we have a peer-reviewed publication that I'll share with you, which talks about recovery times when you actually include an exosome. But it's not really like a one-shot deal. They're using it like 2 or 3 times a day for the days after that procedure. And it'll accelerate that recovery time, by up to 50%. It should. But the idea then is to get them on daily use. So at least with our line, maybe they would start, for instance, with a plated calm. You're introducing them to that product, with some nice cooling agents. But then they would transition to another SKU in the portfolio and use it as daily skincare, to sort of maintain results and preserve that nice collagen and elastin, formation. It's a great question.
So we're kind of counting, like platelets help in the early stages of healing. So they start with that. They work within the hemostasis and the inflammatory component that, or the early stages. So are we counting on the body to continue in the rest of the healing stages? Like the exosomes will kind of get it started, and then the body to kind of go from there on.
Yeah. Yeah. Thank you for asking that. Right. So in a bottle of plated skin science, there's no blood or whole platelets. So it's a different sort of concept. So, after a microneedling or, a laser, the body's going to do its part, right, to get platelets there and start with that coagulation, that early staging of healing. Exosomes are, they're down the road a little bit. Like if you think about that 3-step process, for instance, in whatever wound care or renewing. So in fact, Lori, like on the medical side of our company, we're in FDA trials for wound care, and that's perhaps the biggest division of our company is this application that is sort of racing through that process. So and that's what these exosomes are doing. Yeah.
Excellent.
Yeah. So this is very important as well. So on the medical side, there's a couple of decades worth of publications, peer-reviewed publications. What I'm showing now are the publications, peer-reviewed publications for this cosmetic version on plated skin science, the renewal technology specifically. And anytime something new comes to market, again, you're sort of deciphering, right? And that's what we love about practitioners in the US, aesthetic space is they don't wing anything, right? They go to the research, and you guys are really good examples of this, I know. Before, using things on your patient, it's important to roll your sleeves up and tuck into the data. And again, a lot of our clinics find, and their patients find this very reassuring. This is not like a glittery sort of claim or a new marketing message. There is some major heavy lifting in terms of data behind this product, supporting the results that our clinics and our patients are seeing with plated skin science on a regular basis. So lots of studies going on, and these are studies, IRB approved studies, a lot of rigor here, which is really important. We're going to talk about a couple of these studies specifically, to give you an example of sort of that heavy lifting that goes in to the brand and gives a lot of our customers reassurance. So this is our seminal study. This is an IRB approved study that was done up in Rochester, and the nice thing about this is it's evaluating plated intense, this gold fella, on the front of the image here, alone. So there's no microneedle, there's no laser attached to this. Again, very, very important. If you're trying to decide as a practitioner if an agent is working, you can't do that if there's like 2 or 3 things going on with that patient in a study. So this is just plated intense, and there's a washout period, and this is a controlled study. Okay, we're not presenting you like 1 or 2, before and after shots on a case study that was done somewhere and asking you to make a decision. So this is really nice. So this is 56 patients, age range 40 to 85, and they're measuring various signs of skin aging in this study. And we'll just jump into some of the results. Again, this has been published. So reduction of the appearance of fine lines and wrinkles, and this is 12 weeks actually. We're not advocating, and this was a 61-year-old patient. we're not advocating that this is to replace a toxin by any stretch of the imagination, but just good support, about that collagen and elastin, that message that we talk about. So some of these sort of trickier areas, right, around the sides of the eyes, patients did really well in our study.
Nice.
This is improvement in luminosity. Again, just using plated intents a couple of times a day, 44-year-old patient. And this is at 12 weeks, actually.
Look at the reduction in redness.
I know.
Look at that.
Yeah, and thanks for that. And we have a picture coming up here in a little bit, to show how it does specifically on redness. These patients presented with all sorts of, signs of aging, but that was an area, Lori, where patients did really well in our studies. And here's that example.
Yeah.
So this is just at 6 weeks. So this was sort of the fastest sort of onset of action. Any patient that showed up with the appearance of redness did really well pretty quickly. So if you think about the antioxidant potency of this human-derived ingredient, it's really nice. So I think we're seeing that a bit here. This is visible reduction of brown spots at 12 weeks. And then overall, improvements to the appearance of the skin. And this is a 69-year-old patient.
Yeah.
No procedures going on here, just using this product topically. So it's a versatile agent. Again, I think those photos bore that out. I meant on the slide that had peer-reviewed publications, we actually have a publication that shows all of our histology, which is really important as well. So through punch biopsies, using electron microscopy and stains, we see really nice production of collagen and elastin. So I would encourage you guys or your viewers to take a peek at that peer-reviewed publication. It's really, really impressive and goes to the horsepower here behind this brand. So, a myriad of advantages here. And, we typically get asked, so how do you fit something like this in? Again, there's a lot of really good products out there in the US, a lot of skincare brands that patients have pretty good results with. We have some clinics that will, use this by itself, and that's fine, but we try to stay humble with this. And, a lot of our, a lot of folks that did the research on this still suggest that really nice trifecta is a vitamin C, a retinol, and a good exosome product, a good platelet-derived exosome product. And when you have those three things together in a regimen with this on board, it really optimizes the performance of perhaps your retinol and your vitamin C, which is nice. And potentially patients can back off of some of those other agents. So there's a value message there as well. I think the other important thing to note is that it takes the guesswork out of it as well. If you think about how these things work, you have a patient comes in with, varying things going on. And sometimes it's kind of tricky to cobble together a multiple product regimen. With something like plated skin science, you apply the product and it's going to go in there and figure out, what it needs to normalize, which is nice. So it's a nice sort of layer of security for that patient. Any questions on this?
No.
Okay. The second study we'll touch on was published in the Journal of Cosmetic Dermatology. Oh, and this study is evaluating the use of plated COM post CO2, fractional CO2 laser resurfacing of the face. And, it's a platelet-derived product. So you can make the assumption that it's probably going to help with recovery times. But that's not our style. And, we're not going to rest on that assumption. So we did a clinical trial, a controlled clinical trial on it to evaluate how quickly it would help recovery times and potentially improve overall results. So after the CO2 laser on 18 patients, this product was applied 3 times a day for 30 days versus standard of care like a Stratigel. And we saw some really, really nice results here. So up to 50% reduction in recovery time. And this patient had a really nice result by day 7. I think most patients would be happy with this. The calm has some extra cooling agents in it. The formulation, the pH is a little bit different than some other formulations. So, it provides a, sort of a soothing feel to many patients, after CO2. Also, if you could use it after microneedling and other procedures as well to, to potentially enhance recovery times.
So,
Mhm.
shall we bring me up?
Yeah, please.
I don't know.
Sure.
Yeah, I did my CO2, and I bring this up because I've had lots of people contact me regarding kind of the same thing that happened to me. But what would cause maybe this product to cause a little bit more erythema, a little more,
Reaction.
be a little bit different than untreated side, when we're doing CO2?
Yeah.
Or maybe
Yeah.
somebody being a little more sensitive to it, or
Yeah, I mean, there's a lot
anything.
going on right after
Yeah,
CO2, and
yeah.
it's a good question. In this study, for instance, there weren't any reports of adverse events. And we don't typically hear too much of that, Laurie, but it may depend. Like if that patient has other things going on. In our studies, there was a washout period, right? So it was just CO2 and then calm. They hadn't been on anything prior. And there weren't any other injections or anything, going on at the time of, the calm application. So, I mean, that would be a case by case basis. If it's you that ends up with a bad result, I mean, that probably all that matters. But we didn't see much of it in the studies. Yeah.
Okay, okay. Yeah, no, I think I don't think the outcomes were different. I don't.
Mm.
But my plated side, in all honesty, my plated side was more irritated, a little bit more swollen and reddened than the side I didn't do plated on. And that's what threw me for a loop. So what I didn't know, but overall, my expectation was this would be better, calmer, heal faster. That's what I expected. So
Yeah.
I think my expectations were like, this is going to be calmer, it's going to do better. But overall outcome, side by side, no difference. Like as far as it wasn't more irritated, it wasn't, there wasn't a problem at all with it. But it was kind of odd that maybe, some people might be a little sensitive to it, some people might not be. It might be a calm versus intense. There might be an ingredient in there that might be a little bit more inflammatory, I don't know. Interesting though.
Yeah, no, thank you for sharing that. And, we know that, there's an occasion for that to happen, with our products and, really, many products that you use. So, appreciate you sharing. And, again, we don't see much of that or hear much
Good.
of that,
Yeah.
but
Excellent.
yeah.
Excellent.
So this is the lineup as it stands now. We have plated intense, which is sort of the big gun. It's heavily concentrated. It's a half-ounce bottle. We have plated daily, which is a bit bigger a bottle. It's a little bit of a thicker formulation, a little bit more emollient. That's a full 1-ounce bottle, 30 mLs. Calm, which we just talked about, is also available in a back bar. And then we also have a hair serum, which came out just a few months ago. So, if you think about sort of the mode of action of the intense that we talked about earlier on, the hair serum is loaded with actually a couple of trillion of these platelet-derived exosomes. And what it does is preserve sort of the anagen phase of the hair cycle, leading to the appearance of greater fullness and density. And it's a really nice formulation. It's loaded with some premium scalp and hair conditioners. So really nice for patients to use. It's cosmetically elegant. And we actually have a 9-month clinical study that'll be published in the next few weeks, actually. So we're excited to provide more detail on the performance of this SKU specifically very shortly. But it's been a really nice addition to the lineup, which is
Did they try
fantastic.
this on eyebrows at all?
Yeah, yeah, yeah. We didn't in the particular clinical study. But boy, that would be a home run, right? We should take a peek
Right?
at it. There's no question.
What is the
Yeah.
difference between like your intense daily and calm and hair? I mean, is
Yeah,
there different ingredients? Is it
yeah,
the same amount of exosomes in a pump or something like that? Like what makes it different? Because I mean, at the end
yeah,
of the day, if your base is exosomes, what
yeah.
is it that makes it everyone
Quantity
different?
or
I
other
know.
additives. Yeah.
Yeah, it's a fair question, and it's important. And we've actually resisted the urge to blow this lineup up and have like 20 products because of exactly what you're saying. Like the hero ingredient for all of these is a platelet-derived exosome. So as we bring out different formulations, we want to make sure that there's some variances in maybe the ancillary ingredients that make it better suited for this patient and that. So specifically, Gideon, the product, the gold product Intense, there is 1 trillion exosomes, about 15 billion per pump. It's a lighter formulation, a true serum that absorbs very, very well. We use this when a patient shows up with, maybe multiple sort of signs of aging. Maybe there's some texture issues to sort of get them under control. Moving to the right, daily, a little bit less concentrated, still hero ingredient, platelet-derived exosomes, but it's a thicker product, a bit more emollient. Some younger patients prefer this, or maybe patients with drier skin, for instance, would prefer this formulation, coming from Intense or comparing it to Intense. Calm has a different pH. It's got some soothing agents in it that make it a little bit different. And then the hair, again, highly concentrated with the platelet-derived exosomes, but it's got things like biotin in it. Some of these really nice, conditioners and, for the scalp and the hair. So some of those ancillary ingredients are a little bit different from SKU to SKU. It's a great question.
Okay, cool.
Yeah. Yeah.
Um, let's see. We had some questions. Do you have any information about results for combination of retinol and plated? Um, with
Yeah.
regards to that, and then another one about back bar immediately post treatments for microneedling or some lasers or microneedling RF. I think
That
that's a
calm, I think.
definitely yes.
Yeah. Yeah, yeah. A lot of our clinics have really nice success with their patients using it right after microneedling, or laser. Yeah. From a tolerability standpoint, I think, after CO2, every patient's a little bit different, and it depends on what sort of anesthetic they're on. So it should be tolerable. If it's a pretty aggressive CO2, sometimes the patient may have to wait like a day to start using calm. So maybe the back bar is not the best fit for them. Um, one question we get a lot is about growth factors, actually, and if you don't mind, I'd like to talk about that for just a second.
Yeah.
There's again, there's buzz around growth factors as well,
Yes.
Thank you.
and I think that growth factors sort of really highlights the evolution of this regenerative space. So, what I mean by that is they've been around for a long time, right? And when they first came out, a lot of hoopla, and for good reason, right? This was a nice addition to our tool belt. But if you think about, a growth factor, it's delivering a single message, okay? And it's pinging from the outside of a cell, and it's pinging like grow, 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 grow. These things are very, very different. They work from the inside of the cell, and a lot of people will say like a growth factor. If you think about like the regenerative process as an alphabet, for instance, a growth factor represents a single letter, and an exosome with this whole concert of renewing cues is really the entire alphabet. Another difference is that the growth factor products that are available now, um, they're not naturally occurring growth factors. So they're typically lab engineered, available in super concentrated doses, so almost abnormally high
Mhm.
concentrated doses, something that the human body or the cell isn't used to. So it's like an atomic sort of delivery of this message. Grow, grow, grow. An exosome is very different. a naturally occurring exosome is very different. It's self-regulating. It's not going to let things sort of get out of whack. It's just going to normalize. So it's an important question that we get quite a bit of, you know.
Dan, you brought something up that is near and dear to my heart. I had melanoma about 32 years ago. So I'm very sensitive to anything I put on my patients topically that would cause an exacerbation of cellular dysplasia.
Mm-hmm.
I don't carry any growth factors in my clinic. And I used to have one of the big pharmaceutical companies come in to me and tell me I was crazy for thinking that their products that had lots of different growth factors in them. They have like one that's got two pumps, you pump it together, and there's lots of
Yeah.
different, and there's several companies that have growth factors that you put on every day. My fear is hastening dysplasia of a cell because you don't have control of that. And with these exosomes, you don't have loss of contact inhibition in the cell, correct?
Yeah,
Yeah.
correct.
So you don't have a hastening of like a mole turning into melanoma or
Yeah.
something turning more quickly into a basal cell carcinoma. So with exosomes, you're not going to have to really worry about that as you do with growth factors. So
Yes, for
just
sure.
Yeah.
remember that out there with the
Yeah,
products you're using.
that is one of the things. Yeah, I was at a biohacking conference this weekend, and that was one of the things there, because they were talking about stem cells and stem cell actually injecting. And, there's all this fear that people bought behind it with dysplasias and causing cancers and stuff like that. And their whole thing is the body regulates itself. When you get the whole thing, even though we don't have the exact science of everything, when you look at the totality of it, it really has its own self-regulating mechanism versus if you're just using like a platelet-derived growth factor, exactly one growth factor, and you hypertonic it, you literally just force it through the wall instead of actually having the balance of things that we may not even have all the research on
Right.
that actually have that right balance. And so this is the thing people, as we get into a lot of this, people are like, oh, that works great, as we do and with life, that one thing works great. Let me just take more of it and just keep doing that. And when we're talking about these type of biologics, it really comes back to the balance that our body is always in balance. It's always has its own counterintuitive aspects of it. It checks and balances itself. And so things like this are going to have its own, balanced system because it is a biologic in a more complete form.
Yeah.
That
So
aspect.
I calibrate this watching. I just caution if you're using growth factors topically from any company, you might want to rethink this and look into it and look more at the topical exosomes. Exosomes are, I always say stem cells are like a car, exosomes are like you pull out the engine and you don't have the
Mm.
DNA. You're pulling out the active part, you're pulling out the good stuff that really does the job, and you don't need all the other excess stuff in that cell. So consider changing over to something that would not cause possible skin issues or dysplastic cells to grow.
Yep.
Yeah.
Yeah, that's great perspective, Gideon and Lorian. Thank you for that. And I would even say, like, within the exosome category, remember there's a couple of camps where there's platelet derived and then there's stem cell derived. And I'm not, it's not our style to disparage any of the other companies. They're all great products, and if you're using them, sort of hats off to you. But the reason that our company moved from stem cells to platelets is in a lab, when you use a stem cell, you have to expand that stem cell in the lab setting over and over and over again, that cell line. You dump factors on it, you dump, platelet lysate on it to get it to grow. And, when they grow and expand and separate, divide, they soak up those factors and then they regurgitate sort of the slurry, and that's where they get the exosomes from. But my point is that every time you expand a cell in a lab, you get a little bit of chromosomal damage. We call it phenotypic deviation. So every time you divide, that stem cell it becomes farther away than how it was in the body. And that can lead to, some inconsistencies in terms of performance. Maybe not the nefarious things that we were just talking about, but from a consistency standpoint, that's unavoidable. Platelets are very different. Platelets don't have a nucleus. They don't divide. So every couple weeks, we take platelets up to Rochester. We separate the exosome from the platelet. It's only used once. So what you're ensuring with this sort of fresh sort of source material is this consistent result out of the exosome that comes out of it. So it's a very important distinction, and why at least our company moved away from stem cells 7 or 8 years ago, up in Rochester. So
One of our viewers has a good question. What about PDGF? That's becoming very popular right now. These platelet-derived growth factors.
it's
You see that?
platelet-derived growth factors. Yeah, I
Yeah.
think it's the same thing kind of as we talked about, right? It's and again, if your viewer is getting good results with it and the patients are, certainly have at it. I think uh, per our conversation just a little bit ago, it's very different than an exosome. It's delivering one growth factor, one message. And we know that in the body, sort of that regenerative process is much more multifaceted than that one
Yes.
message. So, there's a balance, Gideon, you mentioned this a little bit ago, that needs to occur inside of that cell to really produce normal, or natural results. It's a fair question. But that's not a naturally derived product. That's a lab engineered product. Very different.
Yes. Yes.
Yeah.
And Emily has a question about the PRP and exosomes. And one thing that's interesting that people confuse a lot of this stuff. Number one, they think PRP gives you volume. It doesn't. PRP is great. It totally depends on the person you're getting from, if they're 20 or if they're 70. So,
Yeah.
there's different health of platelet, ability for the platelet to be healthy. But keep in mind, PRP is very pro-inflammatory, and exosomes have a tendency to be very de-inflammatory. So you're going to see very different results when you're putting PRP versus exosomes with inflammatory components of that patient's response. So two different things you're going to see with that.
It's a good point. So you've activated those platelets in that aggressive spinning, right? You've agitated them. We understand that, and our process is very different. You have to keep those platelets in a very relaxed state all the way through, so they're not producing those cytokines and perhaps leading to inflammation. It's a big component of what we do. But you mentioned it earlier, Lori. The PRP conceptually is a really cool idea, and again, you may get really good results, but it's very reliant on that patient. So if that patient, had a bucket of chicken and a 6-pack of Miller Lite before they came in, I don't know what's going to happen, right?
Very
What this
true.
And if
process,
they're on a truckload of drugs and they have all kinds of
yeah, you never
problems.
know.
Yeah.
So this product, using a pooled donor process, ensures a bit more consistency there. And so we know that our product is firing between, a set of sort of enzymatic guardrails, each lot that's produced. So whether you buy it in January or you buy it in October, it's going to be firing, with that renewing enzymatic activity, to a required degree, which is, it's important.
Excellent. So one of our last questions, we're kind of over. How do people bring this line into their med spa? Melissa has that question.
We're not taking new customers. We're actually at capacity. No,
We're not.
That would be,
Sorry.
that
We'd
would
be
be
like,
amazing.
why are we here? Yeah.
That'd be
you said this was
amazing.
purely educational. No.
I love it. I love it.
Yeah, so we have a team of representatives. Like, heck, we're over like 30 representatives now. Certainly inquire if you don't have local representation at platedskinscience.com. And there's an area where you can inquire, and we can get you in touch with a local representative from the company. But yeah, we're excited about your interest, and thanks for asking that. We'd love to have you and your patients enjoy the benefits of this unique brand.
Excellent. And our team is putting the link up right now so you guys can have the link. I see them typing it in, you guys. There you go. It's platedskinscience.com. Well, thank
Perfect.
you so much,
I
Dan.
know.
This has
Thank
been
you
very,
so much.
very interesting. Thank you so much for giving us all a great education on plated exosomes and where they come from, how they are derived, where, the research that's gone behind this at a very reputable places, and kind of what's gone into this and the outcomes that you can get and all the research. We love the research. We love looking at things that are very research based. So thank you so much for giving us information. We really appreciate this.
It's been my pleasure. Thank you for the time this evening, and have a good rest of your week.
You too. Thank you
Thank
so
you.
much. And thank you everyone for joining us.
Thank you everyone for joining
Dan.
us. We'll see you guys at the next AI
AI
Live
Live.
where we talk about ultrasounds.
Yep, March 4th at
March
5
4th.
p.m.